In each episode of this new series, we'll be speaking to some of the fantastic scientists and researchers behind cancer research, and the theme of each will very much be time. And that's because this year, CRUK celebrates 20 years since its formation. In 2002, the Cancer Research Campaign, or CRC, merged with the Imperial Cancer Research Fund to form the charity we are today. So, what better time than a birthday to take stock and consider how far we've come in cancer research and, crucially, where we go next.
Hopefully we can have a drug, but this is certainly not where we were going, because 20 years ago we didn't have the BRCA story. We had no idea. Not the precision medicine that now, we weren't talking that language 20 years ago.
That was Ruth Plummer. She is a professor of experimental cancer medicine at Newcastle University and an honorary consultant medical oncologist. She's also the director of the CRUK Newcastle Cancer Centre. So there really is no one better to speak to in the inaugural episode of this new podcast series that not only looks back at where we've been over the past 20 years with cancer research, but also where we hope to go next. Now, I spoke to Ruth about how life as a scientist and a clinical trialist has changed over 20 years, how the research environment has changed over 20 years, but also Ruth played an incredibly important role in the development of rucaparib, which is a drug that targets a DNA repair inhibition pathway moderated by an enzyme called poly-ADP ribose polymerase, or PARP to its friends. And that is where we pick up our story. Ruth and I wind the clock back and explore the risks and excitement that surrounded the early days of targeting DNA repair mechanisms of tumours as a cancer therapy. At this stage then, early doors in terms of DNA repair being thought of as a therapeutic target, let's say. What was it like then that, back then, as I understand it, it was always considered very promising, but obviously very risky given the cellular disruption that you were kind of bringing on?
Yes, and I guess maybe it's because of what cancer doctors and drug researchers are trying to do. I mean, it is a very different branch of medicine if you think about it, and I try to explain this to patients when I'm talking about the uncertainties in trials and why quite a lot of drugs fail, is that apart from the infectious diseases doctor, where you've got foreign bugs and you want to kill them, we're the only medics who give a treatment where our aim is really to kill the cells. So you've got human cells that have gone wrong in the human, but if you're going to treat them, you don't just want to stop them. You do actually want them to die. So you're trying to kill cells. So I guess we've always used treatments that are quite toxic. It was considered high risk. I guess it also wasn't known just how many ADP ribosylating enzymes there were. So maybe the full breadth of what you were going for wasn't known, but then, chemotherapy, radiotherapy causes DNA damage. That's what cancer doctors have always been prepared to do, and maybe it's because of what we're trying to do. So it was high risk. It meant that CRUK, the CRC funding in Newcastle, actually did take on a risky project that at the time no company was prepared to do. And then QDOS came along, working as well. The AstraZeneca compound became olaparib, where other CRUK funding has helped along, but the one that actually really started it off was rucaparib, with CRUK/CRC completely behind it as a high-risk project. Now, so I wasn't involved in those early bits because these things do take a huge amount of time. But certainly, as it happened in 2001, just as I became an academic, was when the clinical compound that was clearly the clinical candidate was available. But it was high risk, and the CRUK and committees bought into that and supported the team in Newcastle, and so it was Newcastle's first drug discovery project that this has come out of.
I think I'm right. You were the first person to write a prescription for that early phase 1 trial for a patient.
Well, the first 3 I think I wrote, and somewhere in a file somewhere I've actually got a scan of it because it was a handwritten prescription. Yes, no, I was. The trial was sponsored by the Centre for Drug Discovery, so CRUK sponsored and ran the first-in-human, first-in-class trial. And there were 3 other CRUK key centres on board with us, Oxford, Glasgow and Belfast. And my friends and colleagues there said as we went into the clinic, everybody was a wee bit nervous. And they said, it's all right, Ruth, actually, you can do the first 3 patients if you like.
That must have been a very nerve-wracking time.
Yes, except for there are lots of safety nets. You know, the MHRA had looked at all the data, other people had looked at the data. We weren't doing this blind, ethics committees had looked at what we were proposing. And we put in a really big margin for error on the safety, and it would have let us start, based on the side effects we saw in the preclinical trials, slightly higher. And as investigators, we suggested going in even lower, just in case. So we were being as safe as we could. And I think as is well known, the second person we gave the drug to happened to respond and is still around. You know, so it was safe, but in some ways we were very fortunate that it was safe, because it might not have been. And, you know, sometimes things aren't because of what you're trying to do to treat a cancer.
Has anything changed in the way that these kind of phase 1 trials are done over that time, or is it still kind of a standard thing?
It has changed. I mean, there's 2 things that changed. People, because it was a bit clunky and boring, the traditional phase 1 trial design, which is called the 3+3 design, we've had lots of different cleverer statistical designs. What's happened, which I think if it had happened before would have stood us in better stead with the first PARC trial, is that when we've got to what we think is the dose, we now do what we call an expanded cohort, a bigger group of patients. We don't just stop at our 3 to 6. We do a few more to get a bit more security on the dose and often get some more research samples. That wasn't happening then, and that's a good thing. And there are cleverer and better trial designs now to do these early phase trials. But the huge change is the precision medicine, the molecular profiling. And if I look at the Bobby Robson unit, where we run our early phase trials from at the CRUK Centre in Newcastle, about half of our trials, we're looking for patients with a particular molecular change in the tumour. So we now, within the Target National study that Manchester's leading on, and that study, certainly for us, all patients we're offering as early phase trials to, we're also asking them, are they prepared to give a blood test? And we'll do a circulating free DNA profile on it to see if we can match better to a trial, and to check, you know, are we picking up the right mutations? Are we offering patients what we can? And that's the huge change. And therefore, I think we're seeing more responses. It's great. I mean, a lot of our patients we put on trials don't do well, but we've also got these patients who do. You get long-term responders, and that is great as well for the staff on the unit to see that possibility, rather than the sad stories that you've tried, but you know, all of the time it's a sad story.
This whole stratified medicine approach really speaks to how discovery research feeds into clinical research. And, you know, that's one very clear example where it's like we've understood why there's differences in response, and we can actually now begin to do something about it.
I mean, when I'm talking to patients, I explain to them that, you know, to find a new cancer drug, it starts with the scientists looking at cancer cells in this, trying to work out what has gone catastrophically wrong. Why are they growing out of control? Why can they spread about the body? Again, because we know when people are developing, cells move about a bit and line up to make long bones and things, but then they should stop doing that. So what's gone wrong, and can you then target that, is actually how you find new drugs now. They all love the stories about the natural poisons like the taxanes from yew trees and things that are really powerful drugs and modifying those. But actually it's the precision medicine or the understanding the tumour drivers.
So this was right at the beginning of the story where the first initial phase 1 clinical trial started. Mechanistically, what did we know then? Did we know all we know now, or did we in some way have medicine before we really understood what all of this mechanism was?
No, we absolutely did. And that's the bit of the PARC story that gets lost, in that we had started, as had by that stage some other companies, looking at PARC. Chemotherapy and radiotherapy cause DNA damage, but it gets repaired, and that's a cause of resistance. This was supposed to be a way to overcome resistance and improve existing treatments, which is why we did a combination study to start with. So that's what we were trying to do. And actually, the CRUK compound should have gone into the clinic in 2001, but it for various reasons, and we then ended up with a better, more potent compound, went into the clinic in 2003. The story that's really driven this forward, and it's where the science has absolutely informed this, is that synthetic lethality with the BRCA, that actually wasn't done and published till 2005. And we, by that stage, already had identified that we had a PARP inhibitor, it was safe to give to people, and we knew a dose that blocked the enzyme. So the clinical stuff was moving on, but then the science moved on, and we adapted. So within 6 weeks of that, the New Agents Committee for CDD had approved our proposal to do a phase 2 study in BRCA, breast and ovarian cancer, to test the hypothesis. So we were adapting to the science because that was still progressing.
So 20 years ago, did you think we would be here with it? Obviously, you were hopeful that it would be fruitful and generate an actual drug, but did you think we'd get here?
I was hopeful we'd have a drug, but this is certainly not where we were going, because 20 years ago, we didn't have the BRCA story. We had no idea. We hoped we might be successful.
Get to the next
going to make chemotherapy more effective for patients. That's what we were trying to do. Not the precision medicine that now. We weren't talking that language 20 years ago. At medical school, more than 20 years ago, this was definitely not taught.
I have
So that pretty much
spent
brings
a
us
bit time on the face and the head.
And for those that aren't entirely au fait with DNA repair mechanisms and how they can be used as a therapeutic target, there are links below in the description that take you to a couple of different articles that tell this story and point out the links between BRCA and PARP.
By the way, this is an old episode that I'm listening to if
That, of
anybody's
course, is not the
wondering.
end of the PARP ride. As clinicians refine the use of PARP inhibitors in patients with BRCA mutations, and as we increase our ability to stratify cancer patients, the ambition is that we'll find other tumour types that are responsive to PARP inhibitors. There's also some tantalising hints that the body's immune response to viruses is also involved in the way that PARP inhibitors work. But more on the future of PARP research later. For now, after a career spent on the front line of cancer research, I wanted to talk to Ruth about the working life of a researcher, how things have changed over the years, and what we might be able to do to improve it. So, just before we get back to it, it's probably worth pointing out that aside from her glittering medical and research career, Ruth also has children. So, like many others, navigating a work-life balance has been important to her. So, has much changed on that front?
I think what has changed and is really good is awareness of the talent out there and the stuff that we end up losing out on by making things too difficult. And I acknowledge, phenomenally lucky, partly because the NHS got accommodating people who might want to have career breaks and work part-time quite early, and that's because more than 20 years ago, the evolution of people applying to medical school, it became 50-50 or more female medical students, and then if you've got a limited number coming out at the end and a flow through, you've got a problem in the workforce if you can't adapt. So the NHS is good about part-time and flexible working. But to be fair, I was incredibly lucky with my two mentors, because my kids were quite young when it was suggested to me that we looked at me working school hours. And I thought, I'm not going to survive them being toddlers, let alone school hours. But I did do that. And I talked to my colleagues and they said, well, actually, we're often busier in the morning. And somebody said to me, because he also had small children, he said, we do know if you go home at 3, you're not putting your feet up to watch Neighbours, are you? So you can tell when this was 30 years ago. My husband at the time was on a rotation away from home. I was doing the childcare on my own after work. So, they knew what I was juggling. But I did that for a long time. Even when I was a consultant, supported by the university, that I would have a flexible contract, even when I moved to full time, and would have admin time in evenings, because that was my choice. So I could pick up from school and the children weren't in school till late every day. I was facilitated, and that's what we need. We need that facilitation and the understanding that, well, what was said to me is that if you look at the part-timers, they spend their life feeling that they're not putting enough in and actually putting in more. So it but it's having that flexibility of thinking, and I think that has been a huge change, because I was well supported, but it wasn't the norm, although it was probably easier as a medic, is what I was saying, than as perhaps in a typical science lab where if you had a postdoc who wanted to be part-time, but actually, that's what we need to facilitate. The other bit I think has changed with COVID, and we should keep, much as it would be really nice to come to some real meetings, I think we should keep a blended approach. We acutely discovered we can do things in a different way. And if you want people, the best people on panels, and if you want to interview people, but it might be quite difficult for them to come down to London or wherever you're choosing to have your interview, why can't we use a blended approach? And it's more inclusive. If we just even think of the UK as an island or a series of islands, but it's also more inclusive then of more junior people to bring on the early career researchers, because certainly when the children were little, there's no way I could have gone to London for a meeting. If I would have had to have a whole plan for the evening and look at train being late. We need to meet. I think we are social beings, but I think if we keep a blended approach, it's been one of the few silver linings. You don't want to go back to normal, as everybody's calling it, and think we can lose everything we've gained.
Now, it's time to get back to the science. Time, in fact, to get back to the future, as it were. I set Ruth up with the tricky proposition of telling us where research was headed when it comes to PARP inhibition and other DNA repair inhibitors.
I think it probably is. It's getting them into the optimal combinations, because whether it's loss of BRCA, whether it's germline or somatic, do best. But it's still not everybody, and it's still odd because the biology of the tumour matters. The response rate to rucaparib, for example, in ovarian cancer with a BRCA loss is about 80%. In breast cancer, it drops dramatically to about 40%. In BRCA-related prostate or pancreatic cancer, it's down in the 20%. So there is something about tumour biology we don't understand. Therefore, you would be looking for combinations. And I think there is a rationale for that. It's not just that we haven't got good enough drugs, so let's just shove two in together. It's that understanding the science and how do we make them work together. The other bit of the biology of tumours that helps us to overcome is the fact the tumour evolves and changes. And you sort of hope that if you knew exactly what was wrong with it and caught it early, you might be able to get rid of every single cell with an PARP inhibitor. But that isn't when we get patients presenting. The hematological malignancies seem to have a better chance of doing that. We've struggled in solid tumours, and whether that's delivery of adequate drug within a solid tumour mass to every single cell in an adequate dose, what it is, or it's the fact that they maybe evolve a bit more grow a bit more slowly, present a bit later, even if you try and find them early, and therefore there's more clonal diversity, and therefore it's still tougher. But so I think there are very few cancer drugs that clinically have remained as a single agent. The one that stands out, unless you count radiotherapy as a single agent cancer drug, is something like imatinib in CML. Almost everything else ends up finding its niche as a combination, and I think that is because of the nature of the beast we're trying to treat, to be honest. It evolves, it changes, we don't catch it clonally early enough to get rid of it, and you do get the super responders, and there's a huge amount we could understand from those patients where what was different, because that isn't what happens to a lot of patients. So what's different? And that's certainly, with immunotherapy, that's when we're starting to see trials with complete response rates, and we don't know how long that's going to last because it's early, but what does that mean as well? I'm very nervous about the use of the word cure, partly because as somebody who still sees patients, that's a really big promise that you're making to somebody that you know that much, but it's never coming back. So I'm a bit more circumspect, but I don't know what these complete responses mean. 20 years ago, we just weren't, occasionally you did, but you didn't really see that. But the IO trials quoted 10 to 15 complete response rate in solid tumours, and we just didn't used to see that. And some of the BRCA PARP trials, you've got complete responses. And that is different. And I think that's the tumours that are absolutely terrible, why do they not respond to anything? But actually, there is the other end of the spectrum. If we could understand why, what was right, I mean, maybe it's just that was a tumour that we got lucky with and gave the right drug at exactly the right time. But there should be something
What actually like ideas from the next
to work out how we might
character
do it better.
and done listening?
So clearly lots of exciting research to be done to fully understand part and other DNA repair mechanisms and how we might fully utilise them in therapeutics. Not least among which are the incredibly important things that we could learn from those patients that have a complete response with some of these drugs, which is of course another example of how discovery research and clinical research can work hand in hand. But there is one more twist in the PARP story, and it just goes to show that whatever the future of cancer research holds, we should always be ready to embrace the unexpected. And so I will leave the words. Thank you so much for listening. I hope
Anywhere.
see you next episode.
The exciting stuff now in PARP goes back to people understanding the science and the stuff that was done looking at the fact that if you try to look at a signature in patients who responded, they kept getting this viral signature. What it looks like is if you block DNA damage with PARP, you get bits of broken DNA because it's not being repaired. And within the cell, bits of broken single-strand DNA might look a bit like a virus, which is very much on everybody's mind at the moment, and that stimulates an interferon viral response. So that's why lots of the PARP inhibitors at the moment are being combined with immunotherapy. It's a lateral thinking or a Knight's move bit of science. And now that's very accepted, but it was just completely from off-beat, an unexpected finding that's really powerful in the clinic. And you're then using PARP to get more responses to immunotherapy these days.
Hello and welcome to Cancer Research Matters, the new podcast from Cancer Research UK aimed at our research community. My name is Phil Prime, and for each episode of this first series, we'll be speaking to some of the fantastic researchers behind the work we do. And because this year the charity is 20 years old, what better time to take stock and consider where we've been and where we go next?
The scary thing is, if I'd have done that Sunday afternoon experiment and used circular DNA instead of linear DNA, which I just had in my whole career, would have been different.
That was Professor Steve Jackson, Head of Cancer Research UK Labs at the Wellcome Trust CRUK Gurdon Institute, as well as being the Frederick James Quick Professor of Biology at the University of Cambridge. Now, Steve's main research field is that of DNA damage repair, which 20 years ago was something of a burgeoning discipline when it comes to cancer research. But it is, of course, an avenue of research which has gone on to yield incredible impact when it comes to developing drugs. And speaking of developing drugs, Steve is something of a serial translator with several spin-outs to his name, and so he has an incredible perspective on the last 20 years in terms of how the field of DNA repair inhibition has developed, but also on how scientific entrepreneurship has changed. So settle back and enjoy my conversation with Steve as we take in tips on how to form a spin-out, why the people you work with are just so important, and why his breakthrough in the field of DNA damage repair, like all great scientific breakthroughs, had its share of serendipity.
I actually moved into the field of DNA repair not by design, but
I don't know, I think the name quite speaks for
it goes
itself.
all the way back until basically the late 1980s, when I was working as a postdoc in the San Francisco Bay Area at UC Berkeley. And I did I'm not sure if it was a Saturday afternoon or a Sunday afternoon experiment. It's one of those experiments that wasn't my main thing. And to cut a long story short, I identified an enzyme which we call DNA PK. It's the DNA-dependent protein kinase. And I found it was activated by DNA. And the lab was focused on transcription, genes to RNA, and so published a very nice paper on DNA PK and its potential roles in controlling transcription. A couple of years later, in 1991,
I might actually have to finish finished after this.
in Cambridge, and one of the projects was
Just
to
a couple of long enough.
DNA PK. And within a couple of years, we figured out the reason for an observation I'd made a few years earlier when I initially come across this enzyme. I found it was activated by linear pieces of DNA, but not by circular pieces of DNA. And it took a while for the penny to drop, and I realised that this enzyme, DNA PK, was activated by DNA ends, breaks in DNA. Scary thing is, if I'd have done that Saturday or Sunday afternoon experiment and had used circular DNA instead of linear DNA, which I just had,
Hey.
I never would have made that discovery, and my whole career would have been different. But I did use linear DNA, and the penny dropped. DNA PK is activated by DNA breaks. It's still
Unfortunately,
this was suggesting to me that it was maybe going to be activated by broken DNA.
Do not know you, and I do not allow people to pop up that I don't know.
So my lab was in the early 90s.
I do
focused around transcription,
apologise.
and we were actually making pretty good progress there as well. But in the late 90s, I basically switched off the transcription work in my lab, and since then have just about entirely focused on DNA repair. And it turns out that
And especially when you
the components
just coming in and you've requested to hop up straight away. It's really annoys the shit out of me, that.
Most strand breaks that arise in human cells after radiation, after certain chemotherapeutics that are used in cancer.
That is, let's say that's the glasses. Done.
It was a tiny cottage industry at the time in terms of numbers of groups. And there are hundreds, if not thousands, of groups working on DNA repair throughout the world now. And I can't tell you I started the field.
Sure, so yeah.
The field itself started earlier with researchers such as Thomas Lindahl, who was a very eminent Cancer Research UK funded scientist who was at the co-recipient of the Nobel Prize for that work. So I think one thing that really has changed over the last 20, 30 years is areas of science that were very small.
Actually, realize problem now. I'm gonna have to raise this.
Matured into these much more dynamic, complicated, and rapidly moving domains.
Yeah, all the best scientific stories, in my opinion, start with an afterthought of an experiment, and it was a very serendipitous thing, and then all of a sudden you get this question, you don't know how to answer it.
No, they do. I mean, there are these known unknowns and unknown unknowns, and scientists are most excited when an unknown unknown suddenly becomes a known unknown. You suddenly say, wow, what is this thing doing? But before then, you didn't know it existed. But it's hard to plan for those things. Members of my lab, they're looking at a
Might raise it a bit more higher.
You don't know. Tomorrow you might discover something that really does change the world. You're constantly in a situation where you might see something that nobody else has ever seen before.
When you make a finding like that, did your mind immediately jump to, well, here's the clinical applications and this is worth pursuing? Or was it still at that point just curiosity and interest? Who started?
So that's
For me, it was curiosity, interest, and blue skies. I think if you try to think too much about applications at the start, you're likely to miss true novelty because you can only really make plans based on what you already know. So if you want to strike out a new dimension, you've really got to be, in most instances, thinking in the blue sky space, or at least be willing to act on a chance observation or something unexpected. It was interesting because my first 5 years funded by CRC, predecessor to Cancer Research UK, I scored.
Hey, I might much appreciate it.
But the last statement in the report.
How you doing? Much appreciated for the like.
Because it said basically this all about science. The only question.
Only if I did take my sort ages to get the first one.
And so that did stick with me. A few years later.
Because I'd say that's just layers and layers.
PK enzyme and other enzymes such as PARP, which PARP's polyamide ribose polymerase, that's also a DNA damage activated enzyme. They share that in common. I sort of realised that, these are the types of things that you could develop compounds against, drugs against. I mean, kinases have been drugged, and actually by cloning DNA PK, we learned it was in the PI3 kinase family, for which there were already
So, where are you from then, lads?
molecules, and they weren't ultra selective for DNAPK, but they inhibited it. So initially I was thinking about this in terms of research tools. You could use these to figure out what DNAPK is doing in cells, but then
I know the cell is going to take me absolutely ages, so I know that.
and certain chemotherapy visits. And then the penny dropped that some cells will be affected more than others. Certain cancer cell lines will be more affected than others. And so we didn't really understand why, but the concept of inhibiting DNA repair to selectively sensitize cancers to chemotherapy, radiotherapy, I thought was a really exciting idea. And with that in mind, I founded, with help from Cambridge University, their development office here, and also Cancer Research UK, CRT, founded the company Kudos.
Now, if you'd like the full story on how Steve formed Kudos and how he and his team developed the DNA repair inhibitor,
Because I am actually doing this bit
then
by
I'll put
bit,
a link in the description.
but I'm just
to take you to an article
actually
about
waiting
all of
for the
it. But for now,
paint to dry on the face so I can get a couple of details done on that.
Discovery, a chance finding that opens a
So
whole
I
new
figure
avenue
I'm
of
right
research.
as
It's
well.
about one of the other key pillars, the pub.
Another chance occurrence was it was actually a late night bar at a conference when I was talking to a chap called Alan Ashworth, who at that stage was RDCR UK funded, I believe, at the Institute of Cancer Research, and he'd been working on BRCA1 and BRCA2 cancers for many years with his team. And we had this late night bar. We realised that I, through my Kudos hat, had inhibitors of DNA repair enzymes, and he had some cell lines with and without BRCA1 and BRCA2. So we thought, wouldn't it be interesting if these compounds were selectively toxic to these cells? And within a matter of a couple of months after that conversation, we'd had a few eureka observations, experimental observations, where we found that PARP inhibitors were amazingly toxic to BRCA1 and BRCA2 mutated cells, but not the normal cells. And that identified a clear prediction for how they might be used in the clinic. But as is often the case in science, you can have a great idea, but you're often not the only one with a great idea. And it was after those initial results we realised that Professor Nicola Curtin and colleagues in Newcastle, through their work and communications with Thomas Halliday, who was then at Sheffield University, supported by Cancer Research UK, that they'd also seen the same thing. And so this was, I think, a very good example of coordination. We were competing with each other, but we decided that we would keep each other abreast of developments. And in the end, I think the power of the story was stronger because we published back to back in Nature our findings.
So, transitioning from an academic scientist to someone that is starting and running a company, you're all of a sudden having meetings with venture capitalists as opposed to presenting at scientific conferences. What was that transition like?
I can't say it was a natural or a simple transition. The business world is very different. Although in reality, there aren't very many things to think about. It's all about companies like Good Labs are built on good ideas and good people. And in the end, most companies will only really succeed if they generate something that somebody wants, and in the end, that somebody's willing to pay for. The company is funded to do something, and if it doesn't achieve that, the company's being failure. And that is different than an
Much appreciated for the likes, Sophie.
On one thing, and in 5 years' time, still funded by that original proposal that was successful, you could have gone off in very different directions. And that's great for blue skies academic science. You have to, in a way, see where it goes, how it goes. In companies, of course, you can reinvent yourself or modify your goals. But you do need to get somewhere where you've got materials or products that somebody is going to want to pay for one way or the other. So it was a transition. I was in my mid-30s and I knew nothing about the drug industry or whatever. I was very excited. And so with CI UK's help, I talked to pharmaceutical companies, They weren't interested. Why the heck would you want to inhibit DNA repair? I spoke to VC, venture capital groups, and they had the same initial response. DNA repair is good. Why would you want to inhibit it? In the original Kudos business plan that I developed, there was some text, but there was also a figure showing that if the difference between cancer cells and normal cells is the balance of DNA repair, you may be able to develop drugs against certain DNA repair pathways to selectively kill the cancer cell. And it was that, together with some early data, that convinced some far-sighted VCs to invest in Kudos. It took me almost 2 years with 2 young kids, fixer up a house when we didn't have a roof or any floors or back wall at that stage when I was trying to get the company off the ground. Almost killed me. We got the company funded and we built the team. And the rest is history in terms of it was the world's first DNA repair company. We developed PARP inhibitors, the one that's now marketed as
Well,
olaparib.
then I need to take this down to
As well as inhibitors of a range of other DNA repair enzymes, including
It's
DNA.
handy.
Companies in the world now drugging DNA repair enzymes. So it's nice to see that
Here's
all
the
we start rolling
I need to
is now
pull
rolling
up.
very, very rapidly and in multiple locations.
The idea of being a scientific entrepreneur when I was at university wasn't a thing, or at least I wasn't aware of it. But it seems like a generational thing now. I'm wondering if younger scientists, it's just much more on the tip of their mind because they've grown up with the Jeff Bezoses of the world and they know, oh, that is a pathway to make an impact. So do you think it's become something that's slightly more acceptable or natural for an academic scientist to consider?
I think that is something that's happened. I mean, I did my postdoc in the San Francisco Bay Area, which had the biotech boom before it arrived over here. And I think in those days it was viewed that if you were an academic, let's say, or you do your PhD and then moved into industry, you're selling out or selling your soul, and you're doing dumbed down science. And maybe that did used to be the case some of the time. Now it is much more accepted, expected even, that if you're an academic scientist and you identify something that has translational potential,
Right.
you're expected to
Here's on the video before.
The world is more oriented now than it used to be. But maybe there's more examples of those and people can see that it is possible. You say Jeff Bezos and others, I mean, we're all influenced by good or bad stereotypes and paradigms. The funny bodies now, again, it used to be more difficult to balance the two. Organisations like Cancer Research UK, other charities, it is an expectation now that this science, the fundamental science, might give rise to translational opportunities. And the biggest travesty is, I think, if they're not followed upon and translated. And so that has changed. Things also move more quickly now, in all ways, including in the drug discovery process. And now we're in a better position than we were 10, 20 years ago to have greater prospects of success by tailoring the therapies or even the clinical trials in the areas where they're most likely to work. So all these things, the human genetics and genetic testing, DNA sequencing, all these technologies are improving our ability to move quickly and in many ways more smartly than we did 20 years ago.
So amongst all of
Yes,
these
actually
research
see
advances
that.
have impacted on cancer science over the last 20 years, perhaps the clearest example is the completion of the Human Genome Project. Being able to accurately, quickly, and relatively cheaply understand whole genomes is of course vitally important for many areas of cancer research. Now, the Human Genome Project was declared complete in 2003. So, almost 20 years later, has cancer research managed to capitalise on the hype around this incredible effort?
There was hype in that arena, but in the end, it has paid off. It is paying off. It's just taken longer. It's always interesting with retrospectively looking at things that now appear to be hype, whereas at the time, lots of clever people thought it was obvious. Sequencing the human genome doesn't cure disease, but it does provide mechanisms. That's going to help you bring cures. So some things occur much more quickly now than I would have anticipated. Other things have taken longer. It was interesting going back to the Kudos. One of the reasons why it was quite hard to get VC funding wasn't just the counterintuitive nature of in DNA repair, it was the whole idea that this was a boring drug discovery company. It sounded old-fashioned. But there are all these genomics, human genome companies coming up at the same time, many of which had a lot of hot air around them and hype. But in the end, it was interesting that Kudos did develop some drugs. That was sort of through old-fashioned drug screening processes, medicinal chemistry, which has been around for a long time. But in the end, it made it to market through genetically defined cancers from people with germline BRCA mutations, which is reliant on this DNA sequencing technology, which was developed just down the road here in Cambridge. So it's the juxtaposition of those two domains which allow that and other PARP inhibitors and other anti-cancer medicines to make it to market by going into this segmented population of patients, get registration, and now we're seeing PARP inhibitors and other DNA repair inhibitors spreading out into other cancers, including sporadic cancers, which have similar fundamental mechanisms operating, which can be identified by screening, let's say, prostate cancer patients for defects in DNA repair.
Let's talk a little bit about where you see it going next. So, resistance was something I wanted to ask you about. There are some really interesting stories around how you're getting over that resistance, and that's leading on to a whole other area.
Yes, so for me, there are very few therapies that don't come with downsides or resistance in the case of cancer. Cancers are
How is everybody doing tonight?
this is the case for PARP inhibitors as for other therapeutics. Resistance is something we need to bear in mind. And what we're learning in our research in DNA repair is that almost any change that might give a benefit under certain circumstances, say being treated by this drug, will often come with some kind of liability or vulnerability to something else. That change is in most cases exploitable by some agent. So in the context of DNA repair, for example, we know now that if you take BRCA1 deficient cells, at least in culture, and you can downregulate something such as 53BP1 or the shielding complex, which gives resistance to PARP inhibitors, those double mutant cells or those resistant cells still with the BRCA1 mutation but have changed something else, they're stressed in other ways and can often be hypersensitive to DNA cross-linking agents, for example, which are used in the clinic. So it's fascinating mechanistically to follow these resistance pathways and collateral vulnerabilities, but potentially it's useful therapeutically as well, because you could think of, if we can anticipate a resistance mechanism, maybe we can head it off in the first instance by using combination therapy, or if cancers do evolve to obtain resistance to a certain drug, and you understand that, then it may be possible to evolve the therapy to exploit that resistance mechanism. So I think that's a very exciting arena. The complexity with late stage cancers is that you may have many different things happening in different cells, and there might be different mechanisms of resistance. So for me, the ultimate place that I hope we'll get to in another 10, 20 years, hopefully sooner, is we'll understand resistance mechanisms and how they arise, we'll know how to exploit them, and we'll couple that together with very early detection of cancer, or even detection pre-malignant cells that may already have a BRCA mutation but haven't yet evolved to be a full cancer. And we basically kill these damn things before they even cause a problem to the patient. And I think that is feasible, particularly with these fantastic technologies now based on circulating tumor DNA, and where you can detect markers of cancer without actually knowing where it is. And so you could potentially be looking at efficacy of doing these things even before the cancers become a full-blown entity.
If you had one question related to DNA damage repair that you could have answered in the next 20 years, what would that be?
I'll give you two answers, very different answers. The first answer is, we now know just about all of the key players in DNA repair processes. And there's a small, probably 200, 300, 400 proteins that are connected to the cellular DNA damage responses and DNA repair pathways. And that's a large number. We need to know about how they all work. But what we really are just on the tip of the iceberg is understanding the network of how those proteins how they work together as a network and a regulated network. Cells often don't just do things like this pathway or that pathway. They'll crosstalk with one another, or maybe one will be used in this circumstance, another will be used in this circumstance. So there's that additional layer of complexity, the intersections between the different components, I think is a big one. And so I'd really love us to have this more system-wise understanding of DNA repair. And maybe it's not going to be entirely understandable. It might be the kind of thing that with AI and computationally we'll be able to model and whatever. But that may be very, very exciting from a mechanistic point of view, but also from predictive point of views in terms of maybe suggesting new combination treatments for cancers. My second answer for what I'd really like to know the answer to over the next 20 years is the answer to what is now currently an unknown. So there are plenty of things we don't know. And so I think probably the thing that excites me over the next year will be finding an answer to a question I don't yet know exists. Maybe that's a very silly answer, but that's, I'm sure, is going to be the case.
Yeah, not a silly answer in the least. I'm sure you'll agree. So I hope you enjoyed that session with Steve through his career and some of his thoughts on cancer research as it is and how it might be in the next 20 years. There will be links in the description on the full story of how PARP inhibitors were developed, and also links to information showing how we can help you translate your research. Thank you so much for listening, and I will catch you on the next episode. But for now, I wanted to leave the last word to Steve. After such a successful academic career working alongside Cancer Research UK, as well, of course, as a very successful commercial career with multiple spin-outs to his name, would it be even possible for him to pick a career highlight?
It's reflecting on these eureka moments that DNA PK is activated by DNA breaks, PARP inhibitors, working in this context, and maybe slightly less impactful versions of that in the science over the careers. But actually for me, it's the biggest moments relate to people. And look behind me is basically a whole set of photos of people who've been in my lab over the years with their timelines. And for me now, knowing that many of the people who come through my lab, just about all of them have gone on to do very good things, but many of them are actually now world-leading scientists in the Crick Institute, in Toronto, in Dundee, all throughout the world. And I'm still in very, very good terms with just about all of them, and we occasionally get together. So for me, it's people and helping people along who have networks. Some of them married each other and have kids with each other. In the end, for me, it's about people.
I wonder if you guys want to accept them battle things, the way we call them, or co-hosts.
Hello and welcome to Cancer Research Matters, the new podcast from Cancer Research UK aimed at our research community. My name is Phil Prime, and for each episode of this first series, we'll be speaking to some of the fantastic researchers behind the work we do. Because this year the charity is 20 years old, what better time to take stock and consider where we've been and where we go next?
And then, of course, my dream is that the mass spec is really brought into the field so that, we can start talking about proteomic signatures and not proteins that you can use as biomarkers, but really see how the whole plasma proteome changes in a person over time.
That was the voice of Professor Sarah Zanavan. She's based up at the CRUK Beatson Institute in Glasgow, and as you've just heard, she has a pretty incredible vision for the power of proteomics in a clinical setting. Now, talk of a mass spec-based approach to monitor a patient's proteome in real time should get these sci-fi geeks amongst you immediately turning your minds to the tricorder made famous by Bones in Star Trek. Although I grant you it could just be that I need to update my cultural references. But before we get to that, as tantalising as it is, I wanted to speak to Sarah about her discovery science. Being the very model of a modern cancer scientist, she straddles both the clinical and discovery science worlds and is keen to underpin any clinical work with a deep understanding of the underlying biology. As such, hers is a lab of two halves, with her discovery work being focused on an area which, over the last 20 years, has attracted ever-increasing amounts of attention.
We work on the tumor microenvironment, particularly on cancer-associated fibroblasts, and we want to understand the molecular mechanisms that make these cells pro-tumorigenic, pro-metastatic, and that make the tumors resistant to therapy. And our main focus at the moment is on the metabolism of the cancer-associated fibroblasts, so how we can exploit the metabolism, and the reason behind that is because metabolism regulates epigenetics. So, and it's already known that some of the functions of the fibroblast are of course dictated by epigenetic alterations. And the other reason is because the fibroblasts are highly secretory cells, and some of them produce, for example, a lot of extracellular matrix, which suggests that they may need more amino acid to basically support the production of these proteins. And then if you particularly look at the matrix produced by some calves, they are particularly rich in proline and glycine amino acid residues. And that's actually where we are doing our current research, really seeing whether by inhibiting or removing the availability of these amino acids that they really need to make matrix, we can inhibit the production of extracellular matrix, and in this way alter the tumor microenvironment, for example, to make the tumor more accessible to the immune cells, change the phenotype of the immune cells, and at the same time reduce the pro-invasive function that the cells have, because the matrix support basically these invasive processes. So I would say that this is the major focus. We are also looking at how CAFs interact with other cells in the tumor microenvironment, how they can influence the behavior, for example, of endothelial cells that are the major cell type in blood vessels, how this can impact the formation of tumor blood vessels, or how the blood vessels can interact with, for example, immune cells or cancer cells, because it's something that is very little explored. But then if you look at the tumor, basically most of the blood vessels are embedded in the stomach.
I don't know, that's gonna be a second call.
By fibroblasts, which means that factors released by CAFs can actually influence the blood vessels. So that's the basic biology that my group study, and we are particularly focused on breast and ovarian cancer. Breast is triple negative breast cancer, and ovarian cancer is a high-grade serous ovarian cancer, because of both the tumor types, they don't have a lot of recurrent mutations that are targetable. However, they both have a large amount of stroma, of CAF-rich stroma, that interact with the cancer cells. So the idea is really try to understand how the biology of the CAFs in these tumors, and how we can target them to make the treatments available to the patients in a more effective way. Because as I said, because there are no other, no major therapies against the cancer cells, so we really want to exploit the tumor microenvironment.
So when it comes to the heterogeneous milieu that is the tumour microenvironment, an awful lot of interest has been generated. And when it comes to the cancer types that Professor Zanaban is interested in, it's clear to see why.
When we see patient samples in triple-negative breast cancer, it has been shown that compared to other breast cancer types, triple-negative has a quite extensive amount of stroma. But ovarian is amazing because you can have the tissues from the Balkan surgery, so you can really get quite a big piece of tissue. And sometimes you see that it's like a 50-50, you know, stroma cancer cells is really massive amount. And when patients had already had like a chemotherapy and maybe they didn't fully respond to chemotherapy, you still see a lot of stroma and very few cancer cells, really suggesting that, you know, we should be able to do something.
So that's the what and the why of Professor Zanaban.
Much appreciated for that
But when it comes to
follow
change
and
in the past 20 years,
likes. Much
really
appreciated to you.
So basically, when I started my undergrad studies, you know, it was like you study a single protein and, you know, you try to understand what happens in the cell if you manipulate the protein. But I've always been interested in understanding the whole system. I really wanted to understand
How you doing?
all the
What you
proteins
to?
were
Where you
working
from?
with each other and how they basically were changing in terms of amounts, in terms of post-translational modification to trigger a function in the cell. And at that time, I remember that Matthias Mann published his first papers on
No, I'm just
using mass
adding
spectrometry
sort of a couple of little bits out.
quantitatively at the proteins in a cell. And basically, you could look at thousands of proteins or thousands of phosphorylation sites all at the same time. So and then I said, okay, so that's the, you know, the technology that I want to learn, because potentially you don't have to do any more hundreds of questions.
Yeah, so it's got on the I'm gonna need a second
all the
quote like.
information that you want. And Matthias Mann, he's really a pioneer in mass spectrometry proteomics, so he has developed the technology. Now, you know, you can do proteomics starting from a liquid sample, and then the peptides are ionized, and they go into the mass spec, and, you know, you can identify hundreds of proteins without the need to go one by one through a gel.
Okay, for all those not entirely au fait with the world of mass spectrometry, I've put a few links in the description to this episode to take you to some really interesting articles about mass spec and how it relates to proteomics. But really, the key thing here is that, like so many other areas of science, breakthroughs in the technology behind some of the equipment used can lead to incredible advances in the research itself. And when it comes to identifying with high resolution the components of a proteome, there's certainly been lots of them. But as Sarah explains, one of the key breakthroughs for mass spec was moving away from a way of ionizing peptides called, and if you're not a mass spec person, you will have to brace here, matrix-assisted laser desorption ionization. Phew, a new method.
That's a bit of a mouthful.
Spray ionization. And it was this, coupled with the use of new high-resolution mass spectrometers, that really gave the proteomics field powerful tool it had been waiting for.
The major discovery was actually how to use electrospray ionization to analyze the large molecules with the mass spec.
Okay.
So we're going to be able to ionize.
I'm actually getting somewhere with this.
Being able to analyze, identify them at the mass spec. Then the next step was how do we get proteins out of a gel, because at that time you still have to separate the proteins to make the sample less complex, because of course there are thousands and thousands of proteins in each sample. So Matthias, you know, set up how to extract the proteins from there and being able to put them in the mass spec. And at the time
in a minute.
I joined his lab, it was already the time where we were trying to go from the gel to actually other ways to separate the proteins that didn't require a gel. Something that was simpler, and then you can inject the direct protein mixtures in the mass spec. So what changed at that point is that we were not analyzing anymore protein by protein in the mass spec, but we were analyzing mixture of proteins in each sample. And at the same time, we also developed methods to look at translational modifications, so not just at protein abundance. So you could already start looking at protein phosphorylation in a global manner, and then acetylation came. And nowadays we can look at everything, right? So you have lactylation, we are looking at cysteine oxidation, for the redox stress. So now the field has massively evolved and we can look at a lot of different modifications that before were not possible.
So, it'll come as no surprise that a massive increase in the amount and type of data that you can collect leads to another all too familiar problem. Just how do you get useful, relevant information out of that data?
When I started my postdoc, there were already softwares that identify the peptides from spectra that were semi-automated, I would say. So basically, you were using that database with protein sequences to basically assign to each peptide the right amino acid sequence. But still,
Tottenham Arsenal in the puzzle got matches tonight.
that all the spectra were of good quality. So there was still some very time-consuming step, and I spent a lot of time in the lab, and it was going through it. But
That being said, that is me done.
During the time, you actually automated all
For
the
the
data
data.
analysis, and the data were of very good
Hope
quality.
you're all